Alright, in this video we're going to discuss Oligo Saccharaides, which are chains of monosaccharides between 2 and 10 in length. And oligosaccharides are defined by the types of monosaccharides that build the oligosaccharide and the how those monosaccharides are joined to each other. And we'll discuss both of those in this video. So first, in the previous video, we had introduced hemiacetals and how a linear sugar can form a hemiacetal with itself. To create a cyclic sugar. And so this would be the hemiacetal group. But anyway, there's another group that we need to look at when discussing how two sugars attach to each other, and that is the acetal. It's very similar to a hemiacetal. In fact, we start by taking a hemiacetal and we react that with another alcohol under acidic conditions, so this H+, and so now we have two oxygens attached to this central carbon, and each of those oxygens is attached to another carbon. And so it's like having two ethers groups attached to this same central carbon. And the reason that this is important for sugars is that if the alcohol that joins with the hemiacetal, if this is located on a second sugar molecule, then we'll form a disaccharide. We'll join the two sugars together. So if we take, but before we get to that point, we'll show if we take glucose, cyclic glucose again, so this is our hemiacetal group. The hemiacetal group would be this part of the molecule. If we name this molecule, it would be beta D glucopyridose. And if we react that with methanol, for example, as our alcohol, under acidic conditions, then we can join that methanol, that alcohol, to our hemiacetal to form an acetal. And so now our acetal becomes this group here. That we have like this oxygen that's like an ether because it joins these two carbon atoms. We have this oxygen that's like an ether because it joins these two carbon atoms, and they're both attached to the same central carbon atom. And we call this acetal, or this specific bond here, located between this carbon and this oxygen, a glycosidic bond. And so we'll discuss specific types of glycosidic bond later in the video. So now we'll show a disaccharide. If we take 2 copies of beta-D-glucopyranose, actually no, alpha-D-glucopyranose, so here's our first alpha-D-glucopyranose. We take a second alpha-D-glucopyranose. And what we'll do is we'll combine this hemiacetal group on the first alpha-D-glucopyranose with this alcohol group on the second alpha-D-glucopyranose under acidic conditions again. Then we'll wind up joining the 2 monosaccharides into a disaccharide through this glycosidic bond. And this structure is now an acetal. So this was hemiacetal, now it's acetal. And this bond is in alpha 1, 4 glycosidic bond. And why is it alpha 1, 4? Well, it's alpha because this bond, this hydroxyl that we started with our hemiacetal, was in the alpha orientation, and so it points down. If it pointed up, we would have the beta 1, 4 glycosidic bond. Why is it 1? That's because this is carbon 1, 2, 3, 4, 5, and 6. Because the hemiacetal oxygen comes from carbon 1 of our first monosaccharide, and it joins to the hydroxyl on carbon 4 of our second monosaccharide. So that's why it's a 1 to 4 bond. and the arrow always points from the hemiacetal to the alcohol. And so in this case, our hemiacetal is at carbon 1, and our alcohol is at carbon 4. And so we get this alpha 1,4 glycosidic bond. And the name of this disaccharide is maltose. And so if you were to consume maltose, then an enzyme in your digestive system called maltase would cut this in half to create 2 molecules of glucose, and then you could digest those 2 molecules of glucose separately. We discussed the concept of mutarotation and reducing sugars. This maltose is still a reducing sugar because even though this monosaccharide, our right-handed glucose, is cyclic and attached in this disaccharide, it can still open up into a linear form. And because it can open up in a linear form and reclose into a cyclic form, then it can still mutarotate. And it can still act as a reducing sugar. But if we replace this alpha 1,4 glycosidic bond with a beta 1,4 glycosidic bond, we get a different disaccharide. And so I'll show how that looks. So we'll replace our alpha with a beta. Our hemiacetal would look like this now. We'll still join it to this hydroxyl on carbon 4. Still requires acidic conditions. And so now our acetal group is here. That it has a different shape to it, that this points up instead of down. And so that this bond, because this is in the beta orientation, this becomes the beta 1 to 4 glycosidic bond. This disaccharide is called cellobiose. And unlike maltose, we cannot digest cellobiose. Humans do not have an enzyme allowing them to cut cellobiose in half. So even though it's made of 2 copies of glucose, a sugar we can digest, by combining those glucose in with this beta 1,4 glycosidic bond, we cannot digest it. And so this cellobiose is a component of the polysaccharide cellulose, and that's what helps hold plant cells together because their cell walls are made up of cellulose, which is made up of cellobiose, and why we can't digest like grass. So anyway, we'll look at a few other disaccharides. And the first one we'll look at is lactose. that with lactose, if you remember from previous videos, lactose is made up from galactose and glucose. So this is the D-galactopyranose, and this, it will be D-glucopyranose. So this is our galactose, and this is our glucose. Again, we join these through an acetal group. And this glycosidic bond is another beta 1,4 glycosidic bond. And so this lactose is very similar to the cellobiose that we just looked at, the only difference being the position of this hydroxyl here. In cellobiose, this is a glucose instead of a galactose, and in glucose, this hydroxyl points down, whereas in galactose, that hydroxyl points up. And so just this position of this one hydroxyl group affects our body's ability to digest it. And infants in particular are very good at digesting lactose because they produce large amounts of the enzyme lactase, which cuts these two sugars in half, so then the glucose and the galactose can be digested separately. But of course, as we age, we tend to lose the ability to produce lactase, and many people become lactose intolerant. But anyway, the difference between lactose, which we can digest, and cellobiose, which we can't digest, comes down to this hydroxyl group. But they're both joined by this beta 1,4 glycosidic bond. So the next common disaccharide that we'll look at is sucrose, or table sugar. And sucrose is a disaccharide of D-glucose and D-fructose. Oh, that's not right. And what makes sucrose a little interesting is that we actually join the anomeric carbons of the two sugars together. And so this is the glucose, the D-glucopyranose, this bottom one will be D-fructopyranose, because it's a 5-membered ring, whereas the glucose is a 6-membered ring. So it's pyranose versus furanose. And if we look at the shape here, well, this is beta, and glucose is alpha. And so the glycosidic bond that joins these two sugars together is an alpha beta 1 2 glycosidic bond. And you notice this arrow points both directions now, because we're joining the alpha hydroxyl, this alpha position hydroxyl group of the glucose, to this beta position hydroxyl group of the fructose. So it's both alpha and beta, and it's both 1 and 2, and that because both of these are hemiacetals, the arrows have to point in both directions. And so if we number our carbons, it's 1, 2, 3, 4, 5, 6, and then 1, 2, 3, 4, 5, 6. And so we're joining carbon 2 of the fructose, carbon 1 of the glucose, hence the 1, 2. And so in this case, neither sugar can mutarotate, and this is not a reducing sugar because we've sort of locked up that reducing end of the sugar in this sort of double acetal structure. So that's all of the disaccharides we'll look at. There are larger oligosaccharides with 3, 4, 5, all the way up to 10 monosaccharides. And I won't draw those structures because they get too complex, I think. But like peas and beans contain some of these higher order oligosaccharides. So we have like raffinose, which is a trisaccharide, stachyose, which is a tetrasaccharide, and verbiscose, which is a pentasaccharide. And all of these contain glycosidic bonds that our bodies cannot digest. But the bacteria that live in our intestines can. The difference being that when the bacteria digests these oligosaccharides, they end up producing gas. And so that's what causes people to bloat. Lactose intolerance has a very similar mechanism that when humans become lactose intolerant and they're no longer able to break down lactose on their own, the bacteria living in their guts will start breaking down that lactose instead, and it'll produce bloating and diarrhea. So it's not a lot of fun. But there are medications that can be taken, including Beano and Lactaid. And what these medications do is they contain the enzymes that allow us to break down the raffinose, the stachyose, and the verbiscose, or Lactaid would break down lactose. And so if your body cannot naturally break down these oligosaccharides, you can take a pill that contains the enzymes that will break down those oligosaccharides for you and help reduce the side effects from eating these oligosaccharides. Oligosaccharides are also important because they are oftentimes attached to your cellular membranes. And so if we draw a blood cell, this being a red blood cell, even though I'm drawing it blue, that these red blood cells are oftentimes labeled with complex oligosaccharides. And the different oligosaccharides found on the surface of the red blood cell determines your blood type. And so in A blood type would have a certain mixture of oligosaccharides present on the surface, and a B blood type would have a different mixture of oligosaccharides present on the surface. And your immune system would be able to identify those different oligosaccharides. And so if a person with A-type blood is given B-type blood, the immune system will recognize those B-type blood cells as foreign and attack them, causing an immune rejection. If you have AB blood type, then you have both A and B oligosaccharides on your blood cells. And if you have O type blood, then those oligosaccharides are gone. And so that's why O type blood is the universal donor, because they don't have those oligosaccharides on the surface that the immune system can recognize. And so anybody can accept O type blood. So anyway, that's it for our discussion of oligosaccharides.